Hi everyone. Thanks for joining Thermo Fisher Scientific's webinar on innovations in protein detection and gene expression analysis for SARS-CoV-2 research. Since we are faced with a new challenge to our health by a novel coronavirus associated with acute severe respiratory syndrome, SARS-CoV-2, it is a global effort and research goal of highest priority to investigate how the immune system responds to this virus during the course of infection and to uncover pathways that link SARS-CoV-2 infection to immunological events. Understanding of the adaptive antibody-mediated immune response to SARS-CoV-2 is of particular interest and key to define testing strategies, however, also in view of the development of therapies and vaccines to potentially control and prevent SARS-CoV-2 infection and overcome this pandemic. As summarized in the figure, we actually know that major structural proteins are implicated in SARS-CoV-2 infection and replication, including the envelope, membrane, nucleocapsid, and the most prominent spike or S protein, which mediates the viral entry into the host cell by interacting with angiotensin converting enzyme 2, ACE2 as a receptor. Recent evidence also points to additional factors potentially facilitating the viral entry into host cells. So there are aspects and mechanisms that still need to be further explored in more detail. Taking a closer look, as illustrated on this slide, following replication, release and digestion by antigen-presenting cells, fragmented viral antigens are presented to T cells, which in turn interact and activate B cells that finally secrete SARS-CoV-2 specific antibodies of different classes, including IgM, IgA, or IgG. Some of these antibodies have neutralizing potency and by binding to specific viral antigens potentially prevent SARS-CoV-2 from interaction and entry into host cells. However, still many questions remain to be answered and will require comprehensive testing solutions, including reliable serological assays. In this webinar, I will provide an overview of powerful technologies and novel tools for research purposes to explore each of these stages of host immune response induced by SARS-CoV-2 infection, from detection of viral RNA to SARS-CoV-2 specific antibodies and inflammatory cytokine responses. As one of the world's leading experts on developing and manufacturing immunoassays, Thermo Fisher Scientific offers a portfolio that meets a wide range of needs. From build-it-yourself immunoassays, choosing from a large selection of antibodies, proteins, and other reagents, ELISA kits for single-target protein measurement, and newer platforms such as ProQuantum, highly sensitive homogeneous assays using proximity-based qPCR amplification for protein detection on any qPCR instrument, to multiplexing solutions on the Luminex-based platform for measuring multiple proteins or RNAs simultaneously in one well. In this webinar, I will focus on novel immunoassays to support SARS-CoV-2 research provided on the ELISA and ProcartaPlex multiplexing platforms. For research purposes, two new serological assays are available on the ELISA and on the Multiplex ProcartaPlex platform that enable accurate detection of SARS-CoV-2 specific antibodies and will complement molecular PCR-based tests that are actually the cornerstone for detecting acute viral infection. And in addition, they will provide reliable information on SARS-CoV-2 prevalence and potentially identify individuals with protective immunity against SARS-CoV-2. Combining the spike trimeric structure and total antibody detection with the flexibility of either qualitative or quantitative readouts, the human SARS-CoV-2 spike Ig total ELISA provides one of the best sensitive and specific for detection of SARS-CoV-2 antibodies. Taking a closer look at the workflow of this assay, which is of a typical direct ELISA, using the trimeric spike recombinant protein, a specific viral antigen coated onto the surface of a 96-well plate to bind and detect total antibodies to SARS-CoV-2 in blood, plasma, or serum samples. The antigen-antibody complex is then detected by a horseradish peroxidase conjugated antibody and substrate that finally results in a colorimetric readout within a short incubation time of only 1 hour and 15 minutes. In-house evaluation of the ELISA performance, as shown by the two bar charts, included 39 convalescent PCR positive samples and 160 PCR negative samples. 2 samples from a subject declared PCR positive were found below the set cutoff, however, were confirmed negative in other serological assays, overall highlighting the excellent specificity and sensitivity of this ELISA. In a further screening with control samples positive for respiratory viruses other than SARS-CoV-2, such as influenza A, B, and parainfluenza strains, did not show any cross-reactivity with the spike trimer protein. As shown in the graph, a high control here indicated in red, a calibrator in yellow to define the cutoff, and the low control here indicated in green are provided as part of the assay. Except for 3 PCR positive samples here indicated in orange that could be reconfirmed, all tested control samples here in blue were below the set cutoff, resulting in 100% specificity of the assay. Benchmark testing of the Invitrogen branded ELISA to a competitor assay currently on the market showed discrepant results for certain samples as indicated by the red arrows in the graphs. These included 1 healthy negative sample identified as borderline in the Invitrogen assay, which however was found to be above the set cutoff in the competitor assay and thus identified as false positive. 3 PCR confirmed positive samples that could be verified in the Invitrogen assay, however, were in one case identified as borderline or clearly below the cutoff level and thus false negatives in the competitor assay. So overall, the Invitrogen assay shows more results as expected and therefore superior specificity and sensitivity as compared to the competitor assay. Knowledge of the SARS-CoV-2 protein structure and homology level is crucial to understand how the virus acts and affects the immune system. Among the 4 major structural proteins already mentioned, the N protein primarily regulates viral RNA synthesis, while the spike or S protein plays a crucial role in fusion and entry of the virus into the host cell. As illustrated by the 3-dimensional schematic drawing of the structure, the S protein assembles into a trimer to form the distinctive crown-like appearance and comprises multiple parts: an N-terminal S1 subunit, including the receptor binding domain that specifically recognizes the host ACE2 receptor protein, and a C-terminal S2 membrane fusion subunit. N and S proteins show varying degrees of homology between the other 6 strains of coronaviruses, while the N protein shares about 90% amino acid identity with the preceding SARS coronavirus, SARS-CoV, and about 50% with the Middle East respiratory syndrome-related coronavirus, MERS-CoV. The S protein, and especially the S1 subunit, appears to have much lower degree of homology, making it more specific to SARS-CoV-2 antibody detection. The S and N protein homology is particularly low between SARS-CoV-2 and the 4 listed endemic coronavirus strains, also called common cold coronaviruses, that are to some extent prevalent in the general population, however cause only mild respiratory symptoms. To easily measure RNA or proteins associated with SARS-CoV-2 infection, Invitrogen multiplex assays make use of the Luminex multi-analyte profiling technology compatible with any Luminex instrument to provide a flexible high-throughput solution in a quantitative format. The incorporated branch DNA technology of quantigene-plex assays allow for accurate multiplex gene expression profiling of up to 80 targets per well directly from a blood, tissue homogenate or lysed sample without the need for RNA purification. ProcartaPlex immunoassays for protein quantitation enable the simultaneous detection of up to 65 targets in a single sample of plasma, serum, cell culture, supernatant, and other bodily fluids, requiring only 25 to 50 microliter of sample volume, thus combining the efficiency of multiplexing with the accuracy, sensitivity, and reproducibility of a traditional ELISA. This provides a comprehensive solution for analysis of viral RNA to SARS-CoV-2 specific antibodies, as well as the cytokine milieu in the sample of interest on one instrument platform. Importantly, the multiplex assays can be customized by selecting from a portfolio of more than 150 protein targets for human plus additional ones for other species, and any RNA transcript besides targets and panels specifically designed for SARS-CoV-2 research. The Luminex technology is based on the proprietary microplex and multiplex microspheres that are carboxylated polystyrene beads with a magnetic core that have been dyed into spectrally distinct sets, so-called bead regions, resulting in 100 unique spectral signatures that can be individually identified by Luminex systems. Each unique bead set can be bound by a different analyte through conjugation of a target-specific antibody to distinct beads for protein quantitation, or by using target-specific probes to capture RNAs, in essence giving rise to up to 100 different assays in a single well. Taking advantage of the two instrument lasers, one red laser to excite the bead dye and one green laser to excite the phycoerythrin detection dye, enables classification of the respective bead region and quantitation of the target analyte in parallel. In addition to the SARS-CoV-2 ELISA, the new serology multiplex panel on our ProcartaPlex platform provides a comprehensive screening tool for multiple SARS-CoV-2 specific antigens, allowing to distinguish antibodies to these subsets as well to 6 other coronavirus strains while saving time and sample material. Taking a closer look at the panel design of this 11-plex panel that enables the detection of total antibodies against 4 different SARS-CoV-2 antigens, particularly to N and S proteins and its subdomains that are considered as the most immunogenic, as well as to the S1 subunit of 6 other coronavirus strains next to negative control. The broad epitope coverage is an integral part of this multiplex assay, providing a more complete antibody profile and consequently also increasing the sensitivity and likelihood of predicting immunity status. Among various spike protein domains, the S1 subunit shows the lowest homology between coronavirus strains, allowing to monitor and differentiate between these strains, including the 4 endemic strains of common cold coronaviruses. There is evidence supporting the hypothesis that pre-infection with common cold coronaviruses may provide immunity against SARS-CoV-2. As a T-cell mediated immune reactivity to SARS-CoV-2 has been reported to some extent in the general population, and that is supposed to be caused by prior infection with common cold coronaviruses. This might have also implication in view of vaccine development, and therefore common cold coronaviruses should be considered and also monitored. In a beta test by Dr. Doug Fraser at the Western University and Lawson Health Research Institute, the human coronavirus IG total ProcartaPlex panel was used in stratifying studies of intensive care unit subjects with SARS-CoV-2 antibodies. Blood plasma samples from 14 healthy controls, 14 non-infected ICU subjects, and 14 SARS-CoV-2 infected ICU subjects were profiled at different time points. The box plots show intensities for the 4 different plasma SARS-CoV-2 antigens, and demonstrate a robust immune response over time with significant elevations in SARS-CoV-2 immunoglobulins in SARS-CoV-2 infected ICU subjects as compared to healthy controls and SARS-CoV-2 non-infected ICU subjects already at ICU day 2. And this effect is even more, and this discrimination even more pronounced from ICU day 3 on. Using state-of-the-art machine learning for data analysis based on the complete plasma SARS-CoV-2 antigen response, as illustrated in the TSNE plots on the left, healthy controls here indicated in green could be distinctly separated from SARS-CoV-2 infected ICU subjects indicated in red, with a classification accuracy of 100% on ICU day 3. In table D, the relative importance of each SARS-CoV-2 antigen is listed, showing predominant, however, equal contribution of S proteins and subdomains as compared to the nucleocapsid protein. Similar results were achieved when comparing the complete plasma SARS-CoV-2 antigen responses between SARS-CoV-2 positive ICU subjects and SARS-CoV-2 non-infected ICU subjects on different days, as depicted in the plots on the right, showing accurate stratification and a clear differentiation of these two groups of critically ill subjects, which further underlines the excellent specificity and sensitivity of this multiplex serological assay. In collaboration with Professor Angela Slitt from University of Rhode Island, a quantigene singleplex assay was evaluated for detection of SARS-CoV-2 viral RNA for research purposes. 3 different samples, extracted RNA from nasopharyngeal swabs provided by the Rhode Island Department of Health, with known CT values from qPCR testing, including a sample with low, high, and very high positive qPCR signals, together with a reference material, were used to showcase linearity. By graphing estimated copy number against relative light unit signal, excellent performance corresponding to qPCR results with an R-square of 0.98 was observed. Gene expression analysis can help to better understand the molecular basis that may underlie SARS-CoV-2-induced infection on a transcriptional level. Custom research panels on our quantigene platform include up to 49 targets relevant for studying SARS-CoV-2 infection. As a highly customizable assay, it can incorporate host and virus targets in the same panel, allowing relative gene expression data to be obtained for these targets from the same sample. One way to understand differences in the immune response of a subject is to look at biomarkers. Besides viral RNA, these can include specific cell receptors and surface proteins such as ACE2 and the host cell, as already mentioned. Also immune cells and secreted proteins and soluble factors, including cytokines and chemokines, as well as intracellular proteins implicated in immune signaling pathways. As the immune system tries to expel the virus by recruitment of immune cells into infected tissue, this overactive inflammatory response can cause collateral tissue damage, leading to pneumonia and other respiratory conditions. The cytokine storm or cytokine release syndrome, a hyperinflamed state, is a common complication of various pathological conditions that has also been reported in SARS-CoV-2 infections. High levels of inflammatory cytokines, including interleukins, chemokines, colony-stimulating factors, and others, have been reported to contribute to the symptoms of coronavirus infection and result finally in critical respiratory conditions that can progress to more severe forms, even with fatal outcome. In this hyperinflammatory state, immunosuppression is likely to be beneficial. Thus, there's much interest to analyze the level of a broader panel of immunomodulatory markers, also in the context of SARS-CoV-2 infection, to get a more complete picture of a subject's immune status. With over 700 targets, in vitro ELISA kits provide the gold standard method to measure cytokines involved in pro-inflammatory responses to SARS-CoV-2 infection. In a recent publication as cited here, ELISA kits were used to measure cytokines, including TNF-alpha or IL-6, to better understand immune dysregulation of pneumonia induced by SARS-CoV-2. They could indeed identify a unique signature of immune dysregulation in subjects infected with SARS-CoV-2 associated with macrophage activation syndrome and defective antigen presentation driven by IL-6, which could be in part rescued by addition of a specific IL-6 receptor blocker. To further characterize the host immune response on a transcriptional level, targets of interest for cytokine storm research are also available on the QuantiGene platform, which can be combined with other relevant targets of choice to obtain relative gene expression data from the same well. On the ProcartaPlex platform, several pre-configured immune monitoring panels for human and mouse are readily available, including our largest catalog panel to analyze 65 human cytokine, chemokine, and growth factor targets simultaneously in a single well. In addition, 21 selected highly relevant analytes for inflammatory cytokine profiling are combined in a dedicated cytokine storm panel for your convenience, besides user-defined combination of available targets to develop a unique panel for SARS-CoV-2 research appropriate to your needs. If there is still a target of interest which you can't find in our extensive portfolio, you can take advantage of our custom immunoassay development we provide for all available assay platforms, as well as sample testing services and other custom solutions that help shorten the time from sample to answer and support your research. For practical guidance and protocols to gain more in-depth knowledge on a particular research topic or discover latest research tools, visit our novel resource hub on the website that contains substantial technical content for exploring the field of immunology and beyond. I hope I could give you an insight into immunoassay innovations to unveil the immune response to SARS-CoV-2 infection. Thank you for your interest in this webinar.